Hi folks, welcome back. We are in lab number 5, 5 of 10, halfway there. In this lab, I'm going to hopefully do in just 2 videos. One video walking through example 1, which was an RCBD block design, and the other going through a Latin square. In this first RCBD, the RCBD analysis itself is pretty straightforward, but I'm going to use this as an opportunity to do a full suite of assumptions testing, and this is what you see on the first page of your lab. So let's talk about, in this experiment, we are dealing with mulberries, and I'm going to be reading here from the lab. Mulberries being harvested from different farms, and to help visualize this, I've drawn up a little diagram. What it's saying is that I have 250 berries being harvested from each farm, and these berries are being broken up randomly into 5 groups of 50 berries, and then I have a range of storage temperatures being randomly assigned to these batches of 50. So in this particular case, my experimental unit is a batch of 50 berries, and we in the experiment, we are taking firmness measurements on each of those 50 berries, but then taking the mean. So we are subsampling, but we're averaging the subsamples. That's going to give us a better estimate of the true value of these experimental units, and we can run the ANOVA easily with simply one measurement of firmness per batch of 50. In this particular experiment, farms, the 7 farms, are being viewed as a blocking variable. I'm not actually looking at how one farm is better than another. All I'm doing is assuming that farms, the way farms are run and their management practices, are going to introduce some heterogeneity into the experiment, and I want to be able to remove that variation due to differences among farms, so I can really ask this question about storage temperatures. Okay, so I have pulled up the script. That script is Lab 5 Exercise 1, and I've also brought in the data set associated with that and named it firm.dat. And you'll see we have blocks 1 through 7, those are the 7 farms, and we have temperatures. We have 1, 2, 3, 4, 5 different temperatures, and they are at equal spacings, every 4 degrees, 6, 10, 14, 18, 22, and I have firmness measurements at each of those temperatures. So the full data set has 35 experimental units, 5 temperatures across 7 farms, and of course each of these values is the mean firmness of those 50 berries in each case. Okay, with the data set in, the first thing you want to do, of course, is look at it. It has read in this data set and it thinks that my block or my farm is an integer, which it is not. It is a factor, and it also thinks that temperature is an integer, which it is not. It is a factor. We have levels of temperature, and we have different levels of block. These are categorical variables in terms of running the analysis of variance itself. So we need to tell R that, and that's what these lines 9 and 10 are doing. We're taking block and reassigning it as a factor, and we're taking temperature and reassigning it as a factor. So I'm going to run those 3 lines, ask for structure again, and what you'll see is that now block is indeed being interpreted as a factor with 7 levels, my 7 farms, and temperature is being interpreted also as a factor with 5 levels. Those are the 5 temperature conditions, temperature storage conditions. With that in place, the actual analysis of variance is very, very simple. We're going to run the linear model LM, in which firmness, our response variable, is explained now not by one classification variable, but by 2 separate classification variables, explained by the storage temperature that it was kept at, and also explained by the farm it was on. So we have a treatment variable and a blocking variable. And again, the difference between these 2 things is I'm actually interested in the different levels of treatment. I want to know, does one storage temperature perform better than another? I am not interested in the difference level of block. I'm not interested in asking if farm 2 is better than farm 4 or farm 6 is better than farm 7. Temperature is the objective of the study. Block is an error control mechanism. The only reason this is in my linear model at all is simply to control error, so that we can better ask questions about the performance of different storage conditions. And that's it. Temperature, block, because I have one replication per block treatment interaction combination, or one replication per cell in the experiment, I don't have the degrees of freedom necessary to put temperature times block. If I did that, if I added temperature, the interaction of temperature block to the model, look what I get. R gets very upset. And the reason it gets upset is because I have 0 degrees of freedom in my residuals, because my interaction ate up 24 degrees of freedom. That left me 0 sums of squares and left me no ability to run f-tests. So that is why the interaction is not in the model. And the interaction actually is our best estimate of the true experimental error. So I'll run the model as it was written in the script, and boom, we get some result. From the look of it, it looks like there was a huge temperature effect. There are differences in firmness based on what temperatures they were stored at, and also major differences farm to farm. These are really, really low p-values. Before we would run with these results, however, we want to make sure that we met all the assumptions of the analysis here. And that's what the next part of the script is all about. In order to test assumptions, it's good practice after running the model to grab a couple of things, namely your residuals, your residual values, your predicted values, and in this particular case where we need to run Tukey's test for non-additivity, going ahead and grabbing a squared predicted values. And that's what I'm doing in each of these lines. I'm creating a new column in my data set called resids and assigning to that the residuals of my model. I'm doing the same thing with predicted values. I'm going to create a column called preds, and I'm going to create a column called square preds, in which I take those predicted values and essentially square them. So I'm going to run those 3 lines, and now I'm going to look at my data set. And in fact, I'm not going to look at my whole data set. I'm just going to look at the top of it. I'm going to do head of the data set. And you'll see that my data set is now bigger than it was before. I have my blocking column, my temperature column, and my firmness column. These were the 3 columns necessary to run the linear model. And now in addition to that, I have a residuals column, a predicted column, and a squared predicted column. That's great. Now that I have these residual and predicted columns, columns 4 and 5, these are continuously varying numbers in these 2 columns, so I can create a scatter plot of those 2 things. And that's what this line here is doing. I'm creating a plot of residuals on the y-axis, predicted values on the x-axis, and I'm taking it from the firm.dat data set, and here I'm giving the x and y values some names. Let's do that. And here I get a scatter plot of my residual versus predicted values. And what you want to see here is a nice fluffy amorphous cloud with no particular systematic pattern to it, and that's what we see here. So I would expect, based on this, that once we start testing the assumptions, we are going to have a ANOVA-like error that is normally distributed, homogeneous, independent. But let's go in and ask those statistical questions. That's what's happening on line 30 here. We're going to grab the residuals of our model and ask whether or not those residuals, that is, whether or not the error, is normally distributed by running a Shapiro-Wilk test on it. So Shapiro test of the residual column. It runs down here and we get a p-value that is actually fairly close to 1. In other words, this is a likely correlation, the Shapiro-Wilk correlation. This is a very likely correlation, one that you would find if the data were normally distributed. You would find a correlation this low 83% of the time. So we would conclude that these data actually were randomly sampled from a normally distributed population. So our error meets the assumption of normality. Next thing we're going to do is run Levene's test. I have already installed the package CAR. Hopefully you have as well. So it should be on your computer, but you do need to activate it in this session. You can click the box right here by CAR, or you can run this line library CAR. Either way, I'm just going to click the box and you'll see that it has run the library CAR statement. And I'm going to run a Levene's test on temperature, on my treatment. And what this is going to do is ask whether or not variance is homogeneous across my 5 temperature levels. I can do that, and I get a very, very high p-value. I failed to reject the null hypothesis. This particular data set has error that is homogeneous across my different treatment levels. And all this is important because it then justifies taking the average as the mean square error for testing all my hypotheses. That mean square error or experimental error is representative of all my different treatment levels. My next line here, line 36, I'm also running a Levene test on block. I put this in here not because it is something you would do. The reason you wouldn't do it is because you wouldn't be testing hypotheses about differences among farms. But the reason I do it is to make a very fine point about the fact that Levene's test is only defined for one-way ANOVAs. So even though our particular model in this case for an RCBD has two classification variables, temperature and block, when you run Levene's test, you reduce the model to a one-way ANOVA with the variable that you're particularly interested in. And so this is the line you would run for Levene test temperature. If you wanted to ask about homogeneity of variances among the block levels or among the different farms, that is the particular test you would run. Okay, so we are normal. Normal with a very high p-value. We also have homogeneous variance. So we meet that uniformity requirement with a very high p-value. The last test you would run is this test for non-additivity. And again, this is not an assumption of the ANOVA in the way that homogeneous variance and normality are assumptions of ANOVA. This test for non-additivity is giving you some insight into your system, and it's telling you whether or not your blocks, in this case farms, are acting truly as blocks. Because what you want to be able to say is that the conclusions you're drawing about the effects of storage temperature really don't matter, really don't depend on which farm you are at. If, for example, raising the temperature 4 degrees decreases firmness by 10 units, it doesn't really matter which farm you're at. You do not want to see an interaction between those two variables because your intention is to be able to extend your conclusions about temperature beyond just the 7 farms that were included in the study. Those 7 farms should be representative of a broader universe or population of farms that were not included in the study. So to run the Tukey 1 degree of freedom test, it's an LM statement with the RCBD model. Firmness is explained by temperature and block. And then on the end of that model, you tack on squared predicted values as a regression variable because we are probing the quadratic component of this relationship between your observed values, firmness, and the predicted values. And I refer you again to the Tukey 1 degree of freedom explanation in the lecture notes, and also there's a handout on the course website that explains that a little bit more. So I'm running that. You notice I'm calling this a different name. I'm calling it Firm 1 degree of freedom model, and I run the ANOVA of that model. So let's look at that. You would ignore these first 2 rows about temperature and block. This is not the reason we're running the model. The reason we're running this is specifically to look at the p-value here, 0.21. This is telling us that there is not a significant quadratic component between the predicted values and the observed values. And so therefore, we fail to reject the null hypothesis. It looks as though temperature and block are additive, that they have no non-additive component to it. So that's great. Our blocks are acting as they should. And at that point, you could then go back to your ANOVA table and interpret the response. It looks like there's a treatment effect. You could then dig into those 4 degrees of freedom. In this particular case, because treatments are temperatures at even spacings, probably a trend analysis would be appropriate here to dissect that temperature effect. And that's what you see down below in the script. I'm not going to go through that. You would read the data in again, make sure temperature is classified as a numeric variable, and just as you did in homework, you would create the variables temperature squared, cubed into the 4th, and create a model in which block is there, of course, because it's a classification variable, and then you have the 4 regression variables, temperature squared, temperature cubed, temperature to the 4th, and you would run that. And I'm going to leave the rest for you to look at on page, where are we? We are on page 6. On page 6, I walk through that code a little bit and ask you some questions. So take a little bit of time and make sure those things make sense. And if not, ask me.